Hello everyone, welcome back to my channel Priyanka's Education Videos. So today's topic is the continuation of the last one, which was something about the bacterial reproduction. So in my previous video I discussed about the asexual bacterial reproduction, and in this present video I shall be discussing about the sexual reproduction. So uh, today's video will be about bacterial sexual reproduction. How does it take place in the bacteria? So coming to the sexual reproduction in bacteria, it takes place by three ways, that is by transformation, conjugation and transduction. So let's see the first part, that is transformation. So bacterial transformation is a kind of sexual reproduction which can be actually or precisely called as genetic recombination. So this recombination takes place when the bacterial cell uh, uptakes the DNA from the environment. So the available DNA can be because of some bacterial cell disruption, or it can be due to the lysis of bacteria, due to some bacteriophage infection, or it can be because of any atmospheric factors as well. So because of which the DNA, when it is present in the environment, any other bacterial cell, but here I want to mention that not all bacterial cells can uptake the genetic matter or the DNA present in the environment. Okay, so this particular kind of recombination, which is called as transformation, takes place when the bacterial cell absorbs within itself the DNA from the environment. Okay, the uptaken DNA or the genetic material. So in the subsequent steps, the uptaken DNA or the genetic material which was absorbed or which enters the bacterial cell gets incorporated into the host chromosome. It can get incorporated into the host chromosome, or it can also remain as an extra chromosomal material called plasmid. Okay, the bacterial cells. So as I mentioned earlier, that not all bacterial cells can absorb or uptake the DNA from its vicinity. Only few of the bacterial cells will have or will possess that ability, and those kind of bacterial cells which have got that ability are called as competent cells. So this ability of the bacteria will obviously give that particular bacteria an evolutionary advantage and helps the bacterial cells to survive the environmental changes. So let us see the diagrammatical representation of the process of transformation. So you can see the bacterial cell and its DNA, which is represented in blue color, and the DNA which is coming from outside is being represented in red color. So slowly the DNA from the environment or the vicinity enters into the cell, and here you can clearly see that the bacteria is being mentioned as competent bacteria. So there are two possibilities. So in one of the possibility, what is happening is the uptaken DNA or the genetic material is absorbed or is getting incorporated, or else it can also exist as a extra chromosomal material which is called as plasmid. So this is the simple basic explanation about the process of transformation, a kind of sexual reproduction which actually is the genetic recombination that takes place in the bacterial cell. Okay, I hope it is clear. Now, the next form of sexual reproduction, that is conjugation. So, conjugation is a process of genetic transfer that takes place between two bacterial cells through cell to cell contact. Okay, so it involves the participation of two bacterial cells. In this process, one of the bacterial cell serves as a donor cell which transfer the genetic material, and the other part will be obviously the recipient cell. So the bacterial cell which receives the genetic material from the donor cell is the recipient cell. So the donor bacterium consists of a DNA sequence which is called a fertility factor or F factor. So this fertility factor is responsible for the formation of thin tube-like structure called pilus, which the donor uses to contact the recipient cell. So because of this particular structure, which is called as pilus, these two bacterial cells which are involved in the process of conjugation, they are held together. So the pilus draws the two bacterium together. Okay, thus after they are brought together, the genetic material can be easily transferred from the donor cell to the recipient cell. The genetic material that is transferred is generally in the form of a plasmid, that is the small circular DNA. So obviously it gives the genetic advantage to the recipient cell because of conjugation, because the plasmid generally is consisting of antibiotic resistance genes. This is how the recipient cell will be advantaged by, will get the advantage by, the antibiotic resistance will be carried to the recipient cell. Now, this is the diagrammatic representation of the bacterial conjugation, in which you can see the participation of two cells. One cell is represented as F plus cell because it consists of a DNA sequence which actually encodes the formation of the fertility factor or the pilus, and the recipient cell is represented as F minus cell, and this F plasmid the is the part of the bacteria which gets transferred from the donor cell to the recipient cell. Okay, so the one strand of the plasmid, circular plasmid, actually gets transferred through the pilus to the recipient cell, and later on, upon reaching the recipient cell, the single strand replicate itself and again it forms its own double strand, and in the F plus cell also the plasmid undergoes the replication and again it forms its own double stranded plasmid. So this is how the bacterial conjugation takes place in bacteria in both in F plus, and the genetic material will be transferred from F plus cells to the F minus cell. So the next type of bacterial sexual reproduction, that is transduction, will be explained in the next video. I hope transformation and conjugation are very clear. So thank you for watching.